Welcome to this next lecture on human genetics and biotechnology with the focus on biotechnology techniques. I'm Wendy Nofke and I'll be giving your presentation today. Biotechnology is the use of technology to change the genetic makeup of living things. Some examples of how we've used this in the past were to create pesticide-resistant crops or to create crops with higher food yields. One technique is gene cloning. This isolates and copies genes that can then be used for gene therapy. There are four main steps to this. The first step is an enzyme is going to break the DNA at a specific sequence that you want to isolate to be able to isolate the gene. The enzyme DNA ligase is going to be combined with that isolated gene and a bacterial plasmid. Bacterial plasmids are circular DNA that are going to be independent from the chromosome. The recombinant DNA gets inserted into that bacterial plasmid, and then the bacterial plasmid will insert into a living cell during transformation. The organism is then grown in a way that selects for the transformed organism, and then you create a new population that has the new desired gene in it. Another commonly used technique in biotechnology is PCR, or polymerase chain reaction. Here, copies of the gene or DNA are used to make larger quantities that are available for study or genetic testing. There are three main steps to this process. During denaturation, the DNA is heated. This allows the bonds to be broken and to separate the DNA into its two separate strands. During annealing, the DNA is cooled and the separated single strands of DNA are mixed with short DNA sequences. These new sequences can bond with the DNA strands. During extension, the enzyme is added in to add nucleotides to primers and produce new DNA molecules, each with one of the original DNA strands and then a new complementary strand. This graphic shows the process of PCR occurring. Here you've got the original strand, and it's showing that you take it up to the higher temperature first, then it's cooled, the temperature is raised back up again, and the cycle is repeated. And then new strands of DNA are made, first starting with 1, then having 2, then 4, then 8, and so on. This process can actually be fully automated using a thermocycler now and can allow very rapid amplification of the DNA you want to study using this technique. Biotechnology has a lot of applications in medicine. We can transform bacteria to make human proteins. Things like insulin are already largely made by bacteria. Insulin is useful for diabetics who do not produce their own insulin or who produce inadequate amounts of insulin. Cytokines can also be produced this way, which are immune proteins. With pharmacogenetics, we can tailor medical treatments to the genetic profiles. This is being used in some cancer treatments where you identify what is the genetic profile of that particular tumor and then administer medications that are specifically targeted to that particular genetic profile. With synthetic biology, this involves engineering new functions in organisms from other living systems. There are also many applications in agriculture. Transgenic crops are probably the most well-known of these. These are crops with new genes that make them more useful to humans. This can either delay ripening, delay spoilage of the crops, make them more resistant to pests, and other things that make the crops more profitable for farmers. There are a number of issues that are associated with genetic manipulation. A lot of these fall into the categories of ethical, legal, and social issues. For this particular class, you'll have the opportunity to discuss your views on these in the discussion forum.